Hi everyone, welcome to Dr. Chin Biologies, where you can revise biology for Cambridge IGCSE syllabus. Today, we are going to dive into the super cool topics in biology, how stuff move in and out of your cells. This is happening inside you all the time, and it is essential for life. We will talk about the movements in and out of the cells. Your body is made of trillions of tiny building blocks called cells. These cells, they are always working, and just like your house, they need food and get rid of rubbish. Cells need to get things in and out. The doors of the cell is called as cell membranes. This cell membrane is like a security gate. It is partially permeable, meaning it will choose which molecule can pass through them easily and which molecule cannot. So there will be three ways how the substance move in and out of cell by diffusion, osmosis, and antitransport. What does this gate look like? It is a double layer structure called as a phospholipid bilayer. As you can see, it has two layers. It is also described as slightly free-flowing molecule. Let us study about the structures of phospholipid molecule. Each phospholipid molecule, they are made of two main parts, a head and two tails. The head contains a phosphate groups which carry negative charge. So it will make them become a polar molecule which can interact with the water, which is also the polar molecule. And this phosphate head, it is hydrophilic, means it is water-loving molecule. For the two tails, they are made from the fatty acids and they are hydrophobic, which refer to the water heading. They are unable to interact with the water molecule. As you can see in these pictures, the inside and outside of the cell consists a lot of watery fluids. The inside contents of the cell, known as intracellular space, it contains cytoplasm. The outside of the cell, known as extracellular space. So since both inside and outside of cell are watery, the hydrophilic heads, which is polar, they will face outwards, interact with the waters. The two tails made from the fatty acids, they are hydrophobic, which is water headings. So they will face inwards to each other, forming a central water heading hydrophobic regions. Other than phospholipid molecule, the cell membranes also consist of embedded proteins. So as you can see, those proteins, they are known as the carrier proteins or channel proteins. They are embedded within the phospholipid bilayers. These proteins will help to transport certain molecules which are unable to pass through the fatty acid tube. Another thing is they will have this cholesterol. This cholesterol molecule, they are present between the phospholipid. The cell membrane is partially permeable. So it means it will select what particles able to pass through them, what particle not able to pass through them. First and foremost, we talk about simple diffusion. Diffusion is the movement of a molecule from a region of high concentrations to a region of low concentrations. So in simple diffusions, a substance will always move down its concentration gradients, and this process do not require energies and carrier proteins. So in this video, when a drop of orange ink enter the water, so the ink particles will begin to spread out. So they will move randomly from the area of high concentrations to the area of low concentrations until whole solution is being evenly distributed with the orange colors. So molecules are always moving randomly. This is called as the Brownian motions. These random movements will provide the kinetic energy for the diffusions. When a lot of molecules are packed together under high concentrations, so they will bump to each other and then they will spread out to the regions with a very fewer molecule with a low concentrations. So the particle will just move down the concentration gradients, which means they will go from more to less. What are the importance of diffusions of gas and solids? Diffusion is important for us and all living things. It will help the cells to obtain what they need, like the nutrients. For example, the amino acids and glucose, they can move from the intestines with a very high concentration, get into the blood with a low concentrations. Secondly, it can help to get rid of waste products like carbon dioxide. So waste products, they can move out from the cell by diffusions. Thirdly, it can help to carry out the gas exchange for the respirations. You must always remember that diffusion is very important for gas exchange in both animals and the plants. Let's talk about diffusion in the humans. Imagine that when we breathe in the air, the oxygen will enter from the nostril to the trachea and get into the bronchus and enters to our alveolus. So inside this alveolus, they always have a very high concentration of the oxygen. So the oxygen is able to diffuse from the alveolar air space with a very high oxygen concentrations into the bloodstreams, into the blood capillaries surrounding the alveolus wall with a very low oxygen concentrations. So for the carbon dioxide, usually in the blood capillary, they will consist of high carbon dioxide concentrations. So they're able to diffuse from the blood capillary with a high carbon dioxide concentrations and get into the alveolar air space with a very low carbon dioxide concentrations. So when the blood capillary, when the red blood cell inside the blood capillary, they're able to get the oxygen. They will transport the oxygen and get into the target's body cell. So you can see in this video, right? The oxygen, they are diffused from the blood capillary. So as you can see, the oxygen diffused from the blood capillary into the body cell to do the cellular aerobic respirations. And then the carbon dioxide diffused out from the body cell into the blood capillary as a waste product produced during the respirations. Next, we talk about diffusions in the plants. And remember that stomata, they plays a very important role in the plant diffusions. It's because in the leaf, the gas will enter and exit through the stomata. So where are the stomata? Stomata are found in this small hole. You can see, right, this is a small hole which is between two gut cells on the lower epidermis. So diffusions will control the movement of the gas and water vapors in and out of leaf through the stomata. It will help in the carbon dioxide uptakes. So as the concentrations of the carbon dioxide is higher in the surrounding airspace compared to inside the leaf. So usually the carbon dioxide will diffuse from outside and get into the airspace in the miscible cell by the stomata. So later, carbon dioxide will diffuse from the airspace between the miscible cell and get into the chloroplast of the miscible cell during photosynthesis. Then how about the oxygen? It's making the oxygen concentrations become higher inside the airspace between the miscible cell than outside. So the oxygen will diffuse from the airspace inside the miscible cell and get to the outside of the atmosphere. Last but not least, water vapors also diffuse by the stomata pores from the airspace between the miscible cell and gets into the outside air. So this is due to the concentration gradients that cause the water vapors to undergo the simple diffusions to move out of the stomata pores. Now, let's talk about the factors that affect the rate of diffusion. How fast diffusion happens is influenced by few factors. First, let's look at the distance. Diffusion happens much faster when the molecule they have a shorter pathway to traverse. This is actually why the blood capillaries and the lung alveoli are adapted to have a very thin wall, usually just 1 cell thick. So this is to ensure the diffusion distance remains as short as possible for efficient transport. If the blood capillary they have a very thick wall, this will increase the distance that molecule must traverse, causing the rate of diffusion to slow down. Secondly, we talk about temperatures. The higher the temperature, the faster the molecule moves because they have more kinetic energy. The faster moving molecule means they will collide with the cell membrane more often, speeding up the rate of diffusion. In contrast, the lower the temperature, the slower the molecule will move because they have lower kinetic energy.
Moving to the concentration gradients, think of this like a hill. The steeper the hills, the faster you can run down it. In the same way, the greater the difference in the concentrations between two areas. It will causing the particles to diffuse much faster. Just remember that large concentration difference will lead to rapid diffusions. Small concentration difference will lead to slow rate of diffusions. Next, we talk about surface area. If you increase the total surface area, it will increase the rate of diffusions. The cells are adapted for diffusions, like in the villi in the small intestines, root hair cell in the root parts of plants, inner membranes of mitochondria. They have highly folded or extended surface to increase their total surface area. This will increase the rate of diffusions.
Water is important as a solvent in following situations within living organisms. First, dissolved substance can be easily transported around organisms, for example in xylem and phloem of plants, and dissolved food molecule in the blood. Secondly, digested food molecules are in the alimentary canals but need to be moved to the cell all over the body. Without water as a solvent, this would not able to happen. Thirdly, toxic substances such as urea and other substances in excess of requirements, such as salts, they can dissolve in water, which make them to easily removed from the body in form of urines. Water is also an important part of the cytoplasm, and it plays a role in ensuring metabolic reactions can happen as necessary in the cell. Next, we talk about osmosis. You must memorize these definitions. Osmosis is the net movement of water molecule from a region of higher water potentials to a region of lower water potential through a partially permeable membranes. Note there's two important key points here. We only talk about the water molecule and then we use the terms water potential to describe the difference in the concentrations. Means water is moving down its concentration gradient. What is meant by water potentials? Water potential is being classified into high water potentials and low water potentials. In high water potential, in these solutions, you can see there's more water molecule as compared to the solid particles. So water is more than solids, which is in the dilute solutions, like in pure waters or distilled water, with many free water molecules found inside the solutions. In the low water potential, there is more solid particles compared to the water molecule. So this is found in the concentrated solutions, like in the sugar solutions or salt solution, with a very fewer water molecule as compared to the solid particles. So water will always flow down the gradients from the high water potential to the low water potential, pass through semi-permeable membrane. Picture shows the process of osmosis. A membrane divides the beakers into left sides with a high water potentials due to it contains more water molecule than solid particles. And the right side, they are known as low water potential because it have higher concentrations of the solid particles than water molecule. So the left side, they have high water potential. The right side has low water potential. The water able to diffuse from the left side to the right side. So at the end, on the right side, the water level rise. However, in the left side, the water level reduce. So this movement is called osmosis. Osmosis in the plant cell. If you place the plant cell into pure waters with a high water potential compared to the cell set inside the plants with a lower water potential. Therefore, water will diffuse into the plant cell by osmosis. This will cause the vacuum of the plant cell fills up and swell. It pushes the cytoplasms against the cell wall, creates sugar pressures. The cell becomes hard and firm. We say that the cell is turgid now. Turgidity is crucial because it supports the plant and keeps it upright. If you place the plant cell into a highly concentrated sugar or salt solutions, the solutions outside has a lower water potential as compared to the cell set of that plant cell. Therefore, the water will diffuse out from the plant cell by osmosis to the outside solutions. This is because the plant cell they has a higher water potential as compared to the solutions outside. So this will cause the vacuum of the plant cell. They lose their water and then it will shrink. The cell loses its firmness. Initially, the cell becomes soft. We call this flaccid plant cell. Then in the second scenario, if the water continues to leave the plant cell because the solution outside is very, very concentrated, then this will cause the cytoplasm and the cell membrane pull away from the cell wall completely. So we call these states plasmolysis. So as you can see, there's a space between the cell membrane and cell wall due to the cell membrane pull away from the cell wall. So this usually will kill the cell. This is why the plants will wilt and die if you water them with salt water. Plant cells have a strong cell wall, which change how they react to water environments. We look at three states: turgid, flaccid, plasmolyzed plant cell. This picture shows how a turgid plant cell looks like, which is in the healthy state. When a plant cell is placed in pure water with high water potential, this will cause water moves into the cell by osmosis. The vacuum swells and pushes against the cell wall. The cell wall slightly bends outwards. The cell membrane pressed on the cell wall creates turgor pressures. There are no space between cell membrane and cell wall. The cell becomes hard and firm. We call this turgid plant cell. So what is meant by turgor pressures? It is referred to the pressure exerted by waters inside the cell, pushing outwards against the cell wall. This picture showing how flaccid and plasmolized plant cells look like, which is in their unhealthy states. When a plant cell is placed in a concentrated solution with a low water potential, this will cause the water moves out of cell by osmosis. The cell lose their pressure and become soft. We call this flaccid. If the water loss continues, the cell membrane pulls away from the cell wall completely. We call this plasmolized plant cell. It's important to note that the plant cell will not burst. Unlike in the animal cells, like in the red blood cell, which do not contain cell wall, they only have the cell membranes. So it will burst if they are placed in the pure water and there's too much water enters into their cytoplasms. So in the plant cell, they have a very hard, rigid, and strong cell wall. So this cell wall able to withstand the internal pressure built up inside the cytoplasm and limits how much the cell can expand. So this will prevent the plant cell from being bursting if there's too much water enters into the plant cell. Now, let's talk about dialysis tubing, or sometimes called as whisking tubing. It is a non-living, partially permeable membranes made from cellulose. Pores in these membranes, they are small enough to prevent the passage of large molecules such as sucrose, and they only allow small molecules like glucose and water molecule able to pass through the pore of the membranes by simple diffusions or osmosis. First, suspending the tubing in the boiling tube of water for a set period of time. So notice whether the water levels outside the tubing decrease as the water moves into the tubing by the osmosis. So the water will move from the high water potential to the region of low water potential through a partially permeable membranes. Finally, we move into eddy transport. This is opposite of the osmosis. Let us look at definitions. Eddy transport is the movement of particles through the cell membranes from a region of the low concentrations to a region of the higher concentrations. Because particles move against the concentration gradients, so this requires the energy from the respirations and carrier proteins in the membranes to act as pumps. These are examples of process require eddy transports. The root hair cell use eddy transport to pump in mineral ions, for example like the potassium ions, from the soils with a low potassium ion concentrations into the cell sap of the root hair cell with a high potassium ion concentrations for healthy growth. In another conditions, there will be the uptake of glucose by the epithelial cells in the villi of the small intestines get into the blood capillary. This is because in the villi of the small intestines, they usually have a low glucose concentrations. However, in the blood capillary, they will have a higher glucose concentrations. So the glucose will be actively transported from the intestine villi into the blood capillary by eddy transport. Sodium-potassium pump is a specific example of eddy transport found in the animal nerve cells. It requires the use of carrier proteins embedded in the cell membranes to pick up specific molecules. In these situations, they will be picking up the sodium ions and potassium ions. It uses the specialized carrier proteins in the cell membranes to pump the sodium ions out of the cell. and potassium ions into the cell. So basically, the sodium ions will be pumped from inside with a very low sodium ion concentrations to the outside with a very high sodium ions concentrations. And the potassium ion, they will be pumping from the regions of the low potassium ions, which is on outside of cell, into the higher potassium ion, which is in the inside cytoplasm of the cell. So because this movement is against the concentration gradients, which is moving from low to high concentrations, so the proteins actually require the energies which is being produced from the respirations. So the proteins, they will get the energy and then they will able to change the shapes to help to transport the substance across the cell membranes. So later the substance will be released into the cell.